All right, so last time we talked class-wise or for lecture-wise, I know we talked a lot yesterday in lab. I think yesterday was the most I've talked since I've been sick yesterday. And but anyway, we made it okay. So when we were talking in class, we were talking about the bone marrow in general and where it was located. Showed you guys a bone marrow aspiration on YouTube. So we know that is a pretty invasive procedure to do this. And so basically you're putting that, and essentially what it is, it's almost like a tree borer. You guys ever do FFA when you were in school, maybe? Ag or whatever. They have these things that basically you put them up to a tree and you twist them and you can pull a plug out of a tree and you can look at the growth rings. That's essentially what it is. It's just a medical form of that, and you're going in and you're scooping out, cutting out a core and then pulling it out. So where are they doing that at? Even though it's in the iliac crest, it's very similar to right here in this area. Your iliac crest and your hip actually has red marrow all the way through, even as adults. So like where the long bones, like for instance in this bone, this would be a long bone, whether it's I think this is probably a humerus that they've got up there. But this part, as we grow older, becomes yellow marrow. When you're a baby, it's all red marrow all the way through. So you could technically get bone marrow just about anywhere on a child, up to about 12, okay? And it's essentially basically decreases as we get older, and then the red is replaced with yellow marrow. Once we reach about the age of 25, it's pretty much there's more yellow marrow than red marrow, and most of the marrow is found in the proximal ends of our long bones, okay, as we go. So you could get it technically, but the iliac crest, the sternum are the two big places where bone marrow would be collected as you go through, okay? All right, so let's talk about the bone in general, just a little bit, some of the cells that are in the bone that you may come in contact with. We have what's called the endosteal compartment, and this is essentially the site for bone remodeling. Also contains HSCs, which are hematopoietic stem cells as we go. Bone remodeling is done by cells called osteoclasts, okay? There are two forms of bone cells in our body. One is known as an osteoblast, and the other one is known as an osteoclast. The blast cells, just like what we talked about yesterday in lab, the myeloblast, the ruboblast, monoblast, lymphoblast. Anytime you see blast, it's an immature cell or a original cell, okay? So in this case, the osteoblast, which doesn't sound to me, when I blast something, I break it up. But in this case, remember, we're talking about cellular things. So a blast is an immature or a builder, okay? It's a starter cell. So osteoblasts are basically there to produce calcium to ossify the bones or calcify the bones. Whereas osteoclasts are the ones that break the bone down and they reshape it. And this happens a lot, even just naturally in our body. Especially once we get a break, like if we get a fracture or something like that, the osteoclasts go to work a lot more. And osteoclasts are essentially just a weird form of macrophage, okay? They engulf pieces of bone, break it down, and recycle it as we go, okay? The calcium and different things, all right? So this picture is in your book. Basically what this is doing is showing a section of the marrow where bone and those kind of things are formed. The endosteum, you can see that, this area here, and then there is a picture of an osteoclast there, and then osteoblast also. Like we just said, the osteoblasts produce the bone, and osteoclasts kind of remodel it and break it down as we go. But the thing that's important about osteoclast and blast is not only do they work on the bone, but they also produce certain cytokines that play a role in the maturation of white blood cells. Okay, for instance, the osteoblast, okay, which are those early cells, they actually produce cytokines that stimulate the growth of our HSCs into certain cells. So they play a role not only in supporting our bone and making sure we have bone, but also they play a role in the maturation of our blood cells. Okay, you'll see this. We'll talk about this later on. But a lot of times when kids, especially kids, not as adults as often, but kids, when a kid gets leukemia, if you go back and look at their history, their medical history, one of their biggest complaints that usually sends them to the doctor is bone pain. They hurt. Their bones hurt, they say. When they're big enough to talk to you, of course. I mean, you know, there's babies and that kind of stuff. But the reason that is because of the hyperplasia. There's a term called hyperplasia. What's hyper mean? It's what I am, right? I'm hyper. I'm always moving, that kind of stuff. So when we talk about hyperplasia, we're talking about cells. So we're talking about the hyper or the increased production of cells. Okay. So what essentially happens is in our bone marrow, when we have a hyperplasia, okay, so we said already that these bones, essentially, we know this is yellow marrow that gets in here, right? And then we said over here is where our red marrow is, okay? But the red marrow is essentially just cells. That's all it is. It's essentially those hematopoietic cells, whether it's the stem cells, whether it's immature white cells, red cells, whatever it is, okay? So if you had something that was causing hyperplasia, okay, this is going to begin to grow. And if it begins to grow, eventually it moves on down into this area, okay? And essentially what happens is it starts causing the bone to begin to break down. So then those osteoclasts go into overtime to try to fix the bone, okay? Then the osteoblasts go into their way of fixing the bone, producing more bone, calcifying the bone, this kind of stuff. And so essentially what it does, it just causes pain because there's so much breaking and fixing going on in their body, okay? And not only that, the pressure of the bone marrow itself, that red marrow growing, is putting pressure on the bones themselves. And so the kids get joint pain. And of course, you got to be careful. You know, there needs to be some other symptoms. Don't take your kid to the hematologist just because they, you know, they're hurting in their bones, because that's growing, and everything else can cause that kind of stuff too. Okay, but you got a kid that starts running a low-grade fever, bruising easily, complaining of their joints hurting, their bones hurting, those kind of things. Yeah, take them to the doctor, because there's probably something going on with their blood, you know. Usually that's a big sign of leukemia in kids, okay? All right, so one of the things that we're going to talk about, and we've already kind of mentioned it, we'll talk more about it in the next chapter, but is these little areas where cells grow, okay? So this is a kind of a good demonstration, or a good illustration. You have lymphoid aggregates that are usually very close to blood vessels, all right? Not only are they growing close to blood vessels, but they're also growing close to the lymphatic vessels, okay? And the reason is because lymphocytes, the immature lymphocytes, those progenitors, remember we talked about the common lymphoid progenitor cell, they don't hang out in the bone marrow long. They want to get into the lymphatic system so they can get to the thymus and eventually like the lymph nodes so that they can mature, okay? Whereas like erythroblastic islands, which is what is right here, okay? Remember how we talked about there's a macrophage in the middle, and then cells, the immature red blood cells go around them, and they're actually less mature close to the macrophage, and they mature as they go out. They're in their own little niche also, all right? And they need to be close to either, it's usually one of the veins, veins of the bone, so that they can get out very easily into the veins. Whereas white blood cells, for instance, like the granulocytes, EOs, basos, and neutrophils, they're actually usually close to an arteriole because they would rather go into the oxygen-rich environment of arteries than the red blood cells, okay? Now, this is kind of strange, and you know, we'll get to this eventually in just a couple of chapters, but red blood cells, they carry oxygen, but they actually do anaerobic respiration. You know the difference between aerobic and anaerobic respiration? Biology 101. Okay. What's the difference between anaerobic and aerobic respiration?
The presence of oxygen.
Yeah, presence of oxygen. Aerobic, we use oxygen, glucose, break it down, turns it into water, carbon dioxide, and then pyruvate is the byproduct, and then there's several enzymatic reactions, and eventually we turn pyruvate into ATP, okay? Whereas anaerobic respiration, we use CO2, okay? Carbon dioxide is used instead of oxygen to help break down glucose. And so instead of making pyruvate, we make lactate, okay? And then the byproduct of lactate, we can make ATP, but there's a bad waste product called lactic acid. That's the reason, you know, you're fat and old like me, and you work out for the first time, you say, oh yeah, I'm going to turn over a new leaf this year and start working out. You go and start working out, and the next day you can't move because you're sore as you can be. Well, the reason you're sore is because while you were working out, you used up all the oxygen your muscles had available, and it converted to anaerobic respiration instead of aerobic, okay? And then it produced lactic acid, and that lactic acid got stored up in your muscles, and then it hurts, okay? So, but your red blood cells actually do anaerobic respiration all the time, okay? So they don't necessarily have to be close to that arteriole where the oxygen is increased, okay? Of course, they got to have oxygen, but one molecule of hemoglobin only carries 4 oxygen molecules. You know what I'm saying? Now, of course, we have tons of hemoglobin in our hemoglobin molecules in our red cells, but that's to carry to the rest of the body. It doesn't want to use the oxygen inside that it's attached to help itself to live. Does that make sense? It wants to get the oxygen to the tissue. That's its main job, okay? And it has to do with the affinity, and we'll talk more about that later on. But it does. They usually tend to be around an arteriole, the granulocytes do. Whereas the erythroblastic islands, they hang out around veins, which is deoxygenated blood, okay? All right. Let's see. All right, so vascular supply, we've already talked a little bit about this. There's a nutrient artery, a periosteal artery, and basically there's these two arteries enter the bone through small holes. We talked about this. Didn't we talk about this? We talked about this the other day, didn't we? Central vein drains blood from the marrow, takes it back to be re-oxygenated, okay? So what do these guys do? One of the things that they do is they provide a sinus, okay? So this is kind of a weird concept, okay? The other day I kind of lied to y'all just to be simple, okay? I said something about we have a closed circulatory system. Remember we were talking about the heart? And I said you couldn't really have a hole in your heart or you would die. I said we have a closed system. Well, technically, we do have a closed system, but when it enters certain organs, it actually becomes an open system, okay? So it's kind of in both worlds. But to be simple, we say it is a closed system. But anytime you see the word sinus in a organ that we're talking about, like we're going to talk about some sinuses in the spleen and in the liver, those are actually open systems that blood is allowed to flow into, okay? And so the blood actually leaves the vessels at that particular time to bathe that sinuses, okay? And then it enters back through some capillaries and actually gets back into the closed system. But the reason it's still considered a closed system is because there's usually some kind of a vascular or like a shell around every organ, a membrane around every organ that keeps you from bleeding to death. Does that make sense? So it is a closed system, but there are certain times that it does have an open system in it, especially in like the spleen and the liver and in the bone marrow, okay? And the reason they're there is because they're providing open spaces for the blood cells to mature and to get back into the vessels as we go, okay? So those sinuses are important for us, and basically it just gives the cavity in the bone more area to produce cells as we need them, okay? Arterioles, okay, so remember from phlebotomy, we talked about we have arteries. Small arteries are called arterioles. Then you have capillaries, and then you have venules, and then you have veins. Remember we talked a little bit about that in phlebotomy? Okay, so an arteriole is essentially just a small artery right before it goes into capillaries, okay? So they radiate outward from the nutrient artery into the endostem, which we know that inside that endostem is where all these little niches and stuff where our cells are going to grow, okay? All right, so what are the sinuses? This is a good explanation of these. So they're supported by abluminal side. What is the abluminal side? What would that be? Ab is a prefix. Means below. Okay? So luminal is the whole of the artery or the vessel. Okay? That's what we call it. And it can be the whole of anything. It's known as a lumen. We know that we have lumen that arteries and veins go through that are in the bone. Also referred to as foramens. It's all the same. Okay? But abluminal side by adventitial reticular cells, and basically they're called reticular cells, kind of their rectangle shape, and these guys provide support is what they really do. Their whole job is to provide support and places for like the stem cell to stick to, those erythroblastic islands to stick to, all these kind of things, and they actually provide fibers for them to stick to. And they're going to play a role actually in getting the blood from the bone marrow back into the vessels, okay? Because we talked about how that they're formed in sinuses, but where do we need blood to be? We don't need it to hang out in those sinuses, do we, all the time? We need it to be in what? Where do you like for your blood to be there, Brian? In what? In my veins and my arteries. That's where I want my blood to be. I don't know about y'all. Y'all can have your blood wherever you want to, I guess. We're in America. But I like for mine to be in my veins and my arteries, okay? Don't want it to hang out in the bone marrow and in these sinuses, all right? So the reticular cells will actually play a role in helping blood cells, and the term for this is called egress. E-G-R-E-S-S. When cells mature in the bone marrow, they need to get into the peripheral blood. They have to do a process called egress back into the veins. And we'll talk about those in just a few minutes, okay? Don't really care if you know about that, okay? This kind of shows you those sinuses, what we're talking about. The peristeel capillaries, of course, are basically where gas exchange occurs, just like that's what all capillaries do, is provide an area for gas exchange, carbon dioxide for oxygen as we go through. But capillaries also can help us to get the cells to egress back into the system. Why would capillaries be better than, say, arterioles for egression? Let's say, well, I think that would be the term that we would use, egression. Okay. Why would capillaries be better for egression than arterioles or even arteries or even veins, as far as that's concerned. I will wait and stand here and stare at you until you come up with an answer. So in phlebotomy, we talked a little bit about veins and arteries and capillaries. I made a big deal about capillaries. The reason they work as well as they do is because of this. What is this? All right. How does gas exchange occur? Pretty simple question. Just a science question. Y'all are all scientists. You want to be a scientist, right? That's what us lab techs are, scientists. So how does diffusion, well, I just told you, shit. Gas exchange works by diffusion, right? Okay. So what is diffusion? What is the definition of diffusion? Movement of particles from what? High concentration to low concentration, right? Okay, and if I remember right, I said, all right, so the blue whale is the largest animal in the world, right? And there's a type of vole, shrew, that is like one of the smallest mammals in the world, okay? Is the blue whale sails bigger than the shrew? Huh?
I'm gonna say no.
Who says no? Who says yes, that the whale's sails are larger? Who's awake this morning? They're actually the exact same size. No matter how big they are, of course, a blue whale has more sails than the little shrew does. But the reason they have to be the same size is because diffusion. Diffusion is a very, very efficient process that doesn't require any energy at all. But it only works across a very short distance. Okay? Like cell to cell. So your capillaries are usually only about 1 to 2 cell layers thick. Okay? Whereas your arteries, they're several layers thick. You know? Same way with your veins. Your veins tend to be several layers thick. So if I'm a blood cell living in the bone marrow and I've got to get inside one of these vessels, does my happy little ass want to try to dig through layer after layer after layer to get in? No, I'm lazy. I want the easiest route that I can do. And so what it does, it goes to these capillaries, okay? And essentially what it does, you have what's called endothelial cells. And we've talked about this, all right? And essentially, you know, for the reticular cells, it can actually, and a lot of times the endothelial cells may have those reticular cells that we talked about kind of attached to them at certain spots. So here, they can kind of squeeze through the reticular cells, okay? And that's one thing that's cool about white cells and red cells and platelets is they have to have the ability to kind of deform, okay? They can't stay that perfectly biconcave disc that they are, okay? They've got to have some flexibility, all right? And the reason they have to have that flexibility is because once they're formed and they're mature, they've got to get through the cracks between the reticular cells, okay? These reticular cells. But they have to go through the endothelial cells to get into the peripheral blood, which is flowing into those vessels, okay? So the only way that they can do that, though, is they have to be able to penetrate the membrane and squeeze through like a 0.3 micron hole as they go through. And then they eventually get into the bloodstream. Okay? So capillaries, arterioles, and venules are where this wants to take place. Okay? The smallest vessels that are possible for them to squeeze through as they go. Okay? All right? So the stroma. The stroma is the supporting tissue of the bone marrow. Okay? And so it forms those trabeculae that we talked about, those holes all through, provides scaffolding for the hematopoietic cells to stick to be able to grow in these areas. And some of the stromal cells actually produce cytokines that even regulate hematopoiesis, which is what we need them to do. Okay? So it plays a huge role. Okay? Three major cell types in the stroma that you need to be familiar with. These are macrophages, okay? Macrophages do lots of things. One of the things that macrophages do that's very important is when a red blood cell, and we talked a little bit about this yesterday in lab, we talked about how it starts with that rubriblast. Okay? And we talked about how that it's nucleated. Alright? And then as it matures, it gets to a certain point down here where it's a little metarubicyte. Oops. Okay? And then eventually it becomes a anucleated biconcave disc called an erythrocyte. Remember we talked about that a little bit yesterday. Okay? So, if this is anucleated, that means it doesn't have a what? A nucleus. So, where did this nucleus go? Well, the cell eventually said, hey, not going to divide anymore, so I don't need any DNA. If I don't have any DNA, I can't make ribosomes. I've got all the hemoglobin, or about 85% of the hemoglobin that I need. So, why am I carrying around this heavy nucleus? So, I'm going to kick him out. So, he kicks it out into the bone marrow. And one of those sinuses, and it's just hanging out. Okay? So, if it's just hanging out, is it doing anything to help us? No, it's not. So, when we have things that aren't helping us, what happens to them? Something has to eat it. What are that? That a girl. Macrophages. So we have macrophages that are essentially what? What kind of cell? What are they when they're in the blood? They're what called what? They're called monocytes, right? And then once they get them, it's the butt cells. Remember our butt cells? Yeah. And so they basically go and they can move. They create like little amoeboid actions, like pseudopods. Okay? And they can go and they will engulf that nucleus and destroy it. So that's one thing macrophages do. Okay? Another thing that macrophages do is they help form those erythroblastic islands that we talk about with the red cells. So you basically have a macrophage that is here, okay, and it produces cytokines that stimulate. So basically, it's not erythropoietin, but it plays a role. It's another type of cytokine, okay? So essentially, we have like the ruboblast here that are always going to be surrounding the cell, but closest to the macrophage. Then as they get more mature, by the time you get way out here, they're metarubes, okay? And then eventually you may still have like some reticulocytes and even mature red cells around it, okay? So they provide that. They're actually referred to as nurse cells. So that's another name for that macrophage that is in the middle of the erythroblastic island. It's called a nurse cell, okay? As we go. So that's what they do, all right? Reticular cells, what do? They're called fibroblasts, okay? So let's think about it. I said blast is what? Immature cell, right? And if it's called a fibroblast, what's it probably going to produce? Fibers, okay? It actually produces fibrous projections to help support the bone, plus provide little niches or little areas for these cells to grow. And then we've got adipocytes, which are basically just fat cells, okay? Fat cells, they took in some oil, some lipids, and those kind of things and became known as fat cells, okay? Why do we need fat cells in our bone marrow, you think?
For support.
For support, yeah. When we jump, keeps us from breaking our legs and, you know, those kind of things. What other reason?
And it's a storage in case we need to break down something.
What do fat cells store? Lipids and something else. It's very major to us. All right, so I'm going to throw something out there. Just about every type 2 diabetic is this. No. So think, But think about the posture or the people in general. Just about every person, there are some exceptions, but the majority of type 2 diabetics are always what? Physically. Fat. That's exactly right. They are obese. That's a key. Why are they obese? Do you think? It's because they sit on a couch and eat Twinkies. No, it's really not. That's not why they're big most of the time. Why are they big?
Because they can't break down those.
They can't break down glucose. And so where does glucose get stored? In adipocytes. Our fat is one of the biggest supplies of energy for us as we do it. Why do you think, why do you think you lose so much weight on a protein diet? Because you've eliminated what in your diet? The carbs, which is, we don't care about carbs as much as we do one particular carb, which is what? Glucose, right? We don't care about starch and all them, except for the simple fact starch is glucose, right? But we care about glucose. So when I take all the glucose out of my diet, all the sugar out of my diet, what other source of sugar do I have in my body? Adipose sites. Adipose tissue. Okay? And so that's another reason why we have lots of fat in our bone marrow, is because we still have to have some carbs. And so we can utilize those carbs when necessary for us. Because do we go on a protein diet and we quit eating carbs, do we want our bone marrow to stop working? Our blood cells? Do we want the red marrow to stop because there ain't no glucose available to do mitosis? Heck no, we don't, do we? And so it is there to use as a reserve also, okay? So adipocytes, which is basically just fat cells, all right? All right, macrophages, 2 major functions, and we kind of talked about this already. Phagocytosis and secretion of the cytokines. I'm getting ahead of myself, I'm sorry, on the slides, okay? So we've already talked about this. Make sure you understand that, okay? Secondary functions serve as the center of the erythroblastic islands. We talked about that. All right, so let me introduce you guys to a couple of things because you're going to see this a couple of times in the book. They stain acid phosphatase positive, okay? There are 2 groups of enzymes, okay? When we get into chemistry in the fall, you guys, if you make it, got to throw that in there. Y'all know that, right? I'd hurt y'all, have your feelings hurt if I didn't. So in the fall, when you get there, okay, we will do clinical chemistry. And one of the things that we talk about is enzymes, okay? And remember, anytime you see ASE on the end of a word, 9 times out of 10, it is a type of enzyme. And enzymes are proteins, okay? There's this big group of enzymes that we refer to as acid phosphatase and alkaline phosphatase, okay? It's not a specific enzyme, it is a group of enzymes. So in other words, there's several different types of acid phosphatase. Does that make sense? Okay. Well, what's cool about these things is they can actually stain. We can use certain stains to test for groups of acid phosphatase or alkaline phosphatase. Does that make sense? And like back before, like even, and this is telling you how old I am, and I don't really feel like I'm old, but I am compared to the technology that we have today. Like when I went through school, and when I went through MLT school, there was no such thing as flow cytometry, you know? So, and I graduated in 96, which I know y'all are thinking, well, hell, I wasn't born in 96. Some of y'all. Was all y'all born in 96? Y'all wasn't born in 96, were you? Some of y'all weren't, were you? Okay? So when I finished lab school in 1996, and I know this is going to sound crazy, there wasn't even DNA that we could do, you know? I mean, there was no DNA test. There was no such thing as PCR. There was no such thing as that kind of thing. Now, we knew DNA and all this other stuff, and we knew about chromosomes and this other stuff, but like down to where we are today, we're testing a freaking virus for its RNA. You know what I'm saying? And we can do it in 20 minutes. You know, you could analyze DNA back in those days, and they would do it for crime scenes. But you know how long it would take to get just a DNA match? A week, 2 weeks, sometimes, depending on where it was at. They were just starting on the Human Genome Project, okay? So that tells you how long ago it was, you know, or how back things were without the technology that we have today. But like for us to diagnose leukemias, we had to use certain stains. That's how we did it. We stained the cells certain ways, and we looked for certain enzymes based off of the color of the stain. And one of them was acid phosphatase and alkaline phosphatase. And so they would stain different colors, and we'd say, okay, that's got to be a macrophage. If we didn't know what the cell was, we could test it for acid phosphatase, and it would stain. We'd say that's a macrophage instead of a tumor cell or whatever. Does that make sense to you? Okay. So you're going to see this in several of these cells. It'll say something about acid phosphatase positive or alkaline phosphatase positive. I just kind of wanted you to know. And when we get into chemistry, we'll talk about it because we actually have lots of alkaline phosphatase, and us guys have acid phosphatase in like semen. Those kind of things. Semen has lots of acid phosphatase in it. Okay? Those kind of things. And so we'll talk more about that later on. All right? So that's a macrophage, this big old cell here. And you can see it's actually engulfed some things. Okay? So this one big old cell used to be a monocyte in your peripheral blood. Okay? Just when they get into the into the tissues. And, you know, bone marrow is part of the blood, but it's tissue, right? And so they change a little bit and start looking a little bit different, okay? And when we start looking at body fluids toward the end of the semester, you'll see how the macrophages don't look like monocytes, okay? They can, but they don't always, okay? But remember, they did originate from that monocyte, okay? All right? Reticular cells. These are located on the abluminal surface of the vascular sinuses, and that's what we talked about earlier. And basically, they send cytoplasmic processes into the stroma, and they provide areas for the cells to grow, okay? One of the things that's unique about reticular cells is they actually produce a certain cytokine called CXCL12, and this is a critical cytokine for maintaining our hematopoietic stem cell pool, okay? One of the things we're going to talk about in the next chapter, I'll mention it, okay? All right? So, logic, all right? We have, well, let's just say this, all right? This is pretty simple. Remember the other day we talked about a millimeter cube the other day in class, and I said it's essentially a cube. If I could draw a cube that was 1 millimeter by 1 millimeter, you know, and then the depth is 1 millimeter. That's a millimeter cube, right? So I said it would probably be about, that's probably too big. That one little dot right there that you can just barely see back there where y'all are at, right? Okay. Inside that one little millimeter cube, in a normal adult, is 4.6 to about 6.0 million, so times 10 to the 6 red blood cells. Okay? So think about how many dots of blood or area that I have all through my veins and arteries and spleen and liver and bone marrow and all those places. So think about how many red blood cells I literally have in my body. Now we're just talking about red blood cells, right? Now I'm talking about white cells, platelets, and those kind of things. So think about all those cells, okay? And they all come from one HSC. Okay? So, logical question. How many HSCs do I have in my body? Alright? Big number? Small number? Alright, who says a big number? Big number. I mean, to me it makes sense, right? You'd have to have a bunch to do it, right? To cover all those cells. And you got to remember, because we hadn't talked about this yet, but a red blood cell, its average lifespan is only 120 days. So every 120 days, a red cell dies. So it's got to be replaced, right? And it's got to come from a stem cell, correct? So my question is, big number or a little number? I mean, say a big number of stem cells. We have just a huge number. I'm talking millions. Millions of stem cells. Who wants to agree with me? Come on now, don't be scared. Over 1 million stem cells. I know no takers. So y'all think I need to go higher? 1 billion? Trillion. Shit, I don't know what's next. Gazillion. I don't know what you get past the trillion. Okay. What do you think, Chloe? Chloe thinks it's 1 billion. You think it's a lot. Just a huge number, right? Okay. All right, what about 100,000? 100,000. At least 100,000, right? What do you think? How many say at least 100,000? 100,000 stem cells in our body. No takers? $20,000 is about all we have in our body at any given time to help us produce trillions and trillions of cells every day. And pretty much we have about 20,000 that last our entire life, from the time we're little to the time we're adults. Okay? And here's the key. Remember I talked about very early in this chapter that these HSCs don't differentiate and they don't divide. They just stay kind of the same, and they cause the chemical signals to go on those daughter cells that they produce. That common progenitor cell that is produced. It's actually, and I didn't tell y'all this, I'm going to tell y'all, I didn't get into as much detail about it as I will. Okay, so essentially what happens is, I kind of lied. Now y'all are thinking, hell, we can't trust nothing he says. It does divide, okay? But it only divides once, okay? It doesn't produce two like we talked about to start with, okay? I threw that out there because it made sense mitosis-wise. But let's get down to what really happens. What really happens is this cell divides once. It actually makes kind of a clone of itself, okay? And it turns into one cell that is known as a it's called a multipotent stem cell, or it's also known as a pluripotent. That's how you spell it. Pluripotent stem cell. Okay? This guy, and they abbreviate it M sometimes, that multipotent stem cell. This guy is the one that actually gets exposed to cytokines so that it can produce that common myeloid progenitor and that common lymphoid progenitor. Does that make sense? And then this guys then go on and do all the division, the differentiation. Does that make sense? So this guy never changes. It creates a clone of itself that's basically a naive cell. It doesn't know, it hasn't ever been exposed to anything. This guy has been exposed to interleukins from like the reticular cells, some macrophages, to keep it as a stem cell. Does that make sense? So that it doesn't ever change and go into one of these over here, okay? This is what happens sometimes, though, in certain leukemias. This cell stops recognizing that it's a stem cell and then goes into mitosis and starts dividing, and then that one divides, and then this one divides, and so forth and so forth. So we get leukemias or tumors and those kind of things. Does that make sense? All right? Yes?
Okay, I have a dumb question.
No, there's never a dumb question.
So the HSC, do we have the same ones like for our whole
Just
life?
about. Yeah.
Like we don't make new ones
Nope.
ever?
Mm-mm.
Okay.
And that's the reason, like stem cell research is a big deal, because the reason that they harvest, a lot of times they'll take stem cells from like aborted fetuses. So when we're in that fetal stage, we actually have lots of stem cells, okay? Because there's so much mitosis that has to go on. But then once we reach a certain age, we're born, and up to about 2, our stem cells start declining, because by that time things are developed and they just need to go through regular mitosis to grow, okay? So we reserve a pool, what we call a pool, to help us when our body gets stressed. So for instance, like an infection, we need more white cells to be produced, okay? So we always keep a pool of hematopoietic stem cells as we go.
So
Okay.
when you're like a baby, some of those stem cells
Okay.
turn into other stuff.
Well,
they don't all do that?
That's what I was
Okay,
fixing to say.
okay.
No, you're good. You're thinking the right way. These are hematopoietic stem cells, okay? So this particular pool of stem cells that we're talking about here are just blood. Does that make sense? These are the ones that we're studying right now. But you have human stem cells that can turn into nerve cells or turn into, bone tissue or, whatever, okay? But I shouldn't say bone tissue, because actually the osteoclast and blast come from one of these two, okay? But this guy, what we're talking about, 20,000, is what produces our blood, okay? You have other stem cells in your body, that blood stem cell, that hematopoietic stem cell, is not going to make you a nerve cell. It's not going to make you an arm or a liver, that kind of thing. So you have to have other stem cells that do other things. Does that make sense? So maybe I misled y'all. I may have misled you, because I just said I thought y'all understood that. But no, we're talking about hematopoietic stem cells. Okay. just for our blood, what makes our blood. We have lots of other types of stem cells in our body. Okay? All right. These are those reticular cells that you can see there. And they're providing, and you can see they're actually providing those fibers so that you can see the connective tissue that they are. Okay? These are biopsies. The H&E stain is a Hansel, I think, Hansel and Eason stain is what H&E stands for. The H&E is a very common histology stain that they use in lots of histology, like biopsies and this kind of thing. Okay. We talked about adipocytes. Talked about how that red marrow gets replaced with yellow marrow the older we get. And eventually, like we said, by the age of 25, your red marrow is just in certain spots. And this distribution of 1-to-1 in these, that's just in these particular places, okay, that we have. And if you'll notice, the pelvis, the iliac crest is where we get it, the sternum. You can get bone marrow from all these, but like we said before, the sternum and the iliac crest of the pelvis is the two main areas that we would do bone marrow aspirations from as adults. Okay, now babies, kids, they can do them several different places, but even in babies, they like to stick with the iliac crest because it's less tissue there to have to go through, so they can do those. Those are what adipocytes look like there, and this picture's even got a picture of some stem cells. So those little stem cells right there, they look like lymphocytes, and you all see that. A lot of times stem cells look like lymphocytes, but you wouldn't know they were stem cells though unless you did a flow cytometry and looked for certain CD markers on them. Okay, I think CD stands for cell differentiation marker, if I remember right. All right, we talked about osteoblast already. They resemble plasma cells. I've got pictures of you. The only thing that's different about an osteoblast than a plasma cell, the color just about is the same. But notice how that the doning, remember I told about there was a doning where that Golgi apparatus and the endoplasmic reticulum doesn't stain. It's pushed away from the nucleus, okay? In a plasma cell, it'll be right next to the nucleus, okay? And I'll show y'all guys a plasma cell eventually. There's no reason to right at this moment, but I will when it comes time, okay? But these are what osteoblasts. What do osteoblasts do?
Produce calcium to calcify the bone.
They make bone. That's what the definition you need. They're bone makers, okay? All right, the next one we talked about was osteoclast. And osteoclasts are basically modified macrophages, okay? That's what osteoclasts are. So look at that. Multinucleated, big cell, ugly looking cell, mean cell. They break down things, okay? They're bulls in a china shop. Break down foreign things, okay? Bone in particular. When you get those bone fragments from a fracture, they're the ones going to go in and digest them, okay? All right. We talked about niches already. We talked about erythroblastic islands. Talked about granulocytes. Talked about macrophages. Megakaryocyte. We mentioned, I think I screwed up, and that's the reason I hate to do any kind of lecture in the lab is because I have two different groups. Because I know in the second group I talked about a megakaryocyte, right? With you guys and about polyploid and all that, but I don't think I did in the first group with you guys. And see, I have ADD, man, pretty bad, and I couldn't remember what I did in lab 1 and lab 2. So let me do mention this, okay? So in lab 1, if you were there yesterday, megakaryocytes come from that common myeloid progenitor, okay? They get exposed to certain cytokines right here, and that says, okay, I need you to turn into a megakaryocyte, all right? The problem is, megakaryocytes can be polyploid. They can have more than one nucleus, all right? So I talked about yesterday in lab, I said, what's a haploid and a diploid? So what is ploid referring to? You other guys that was in group 1, 2 can tell them. What's ploid stand for? The DNA, said the DNA is part of the what? Come on, Brian, you got it right yesterday, man, you can't forget it. What was we talking about yesterday? You said DNA, and I said DNA makes up our what? Chromosomes, right? And we said that a haploid cell has how many sets of the chromosome? 1. And diploid has 2. So polyploid would have what? Many, right? More than 2, okay? So a lot of times these megakaryocytes, you know, they're going to have they can have up to 4 N. See, 2 N would be diploid, okay? So they can be like tetraploid, you know, or even bigger. So huge source of DNA. If you ever needed DNA, you wasn't real sure about it, you needed it, you could go to bone marrow and try to find hopefully you could get a megakaryocyte and you can get all the DNA you want, right? So basically, the megakaryocyte always stays in the bone marrow. There's one rare, like one in, you would have a better chance of winning the lottery than getting megakaryocytic leukemia. You know what I'm saying? Because there is one rare one, though, that has been documented, that people can get megakaryocytic leukemia, where the megakaryocytes end up in the peripheral blood, all right? But 9 times out of 10, it stays in the bone marrow, where it should always be. So how do we get platelets? Platelets are basically broken off pieces of the cytoplasm of the megakaryocytes, okay? And then they become the platelets, all right? Listen, there's a whole chapter on platelets, okay? So give me time, and we'll get there. When we get to Unit 5, we start talking about coagulation, all right? Lymphocytes, we talked about them just now, where they aggregate. They want to go to the thymus. Hyperplasia, talked about hyperplasia. What did I say hyperplasia is? Increase in what?
The red bone marrow.
Which means increase in what?
Bone production.
Not bone production, but what is red marrow? What did I say red marrow is essentially? Just a bunch of what? Cells. So hyperplasia would be an increase in what? Cell production. That girl. All right. And so you have hyperplasia that occurs. Most of the time you're going to see this in leukemias is usually what happens in leukemias. Okay. You can actually have hypoplasia. Okay. This is known as aplastic anemia, and we'll talk about that later on. Aplastic means no cell production is essentially what it means. So you would see a cytopenia. Have you ever seen that term, cytopenia? Cytopenia. All right, anytime you see the word penia, it means decrease in, okay? So cytopenia would mean a decrease in what? Cells, right? Okay, cell production. So, but then we can go to leukopenia, we can go to thrombocytopenia, and then we can go to erythropenia, which we call it anemia. Okay? All right? So, sometimes that can happen, and you get what's called hypoplasia in the bone marrow. Okay? So, it's a decrease in blood production. Okay? We talked about egressin, put the little drawing on the board where they got to squeeze through. That's how they get back into circulation. Talked about that. Membranes fusing. They got to go through the endothelial cells. This is something that we didn't really talk about. Soluble factors. What's soluble mean?
Dissolvable.
It means it's dissolvable, right? And so they need some help to be able to get through the wall of the endothelial cell. All right. So we have these factors that are cytokines. They're called colony stimulating factors, CSF, okay? Don't get that confused with cerebral spinal fluid, because in the lab we say CSF a lot of times and we're talking about spinal fluid. But when we're talking about hematology, we're talking about a cytokine that's a colony stimulating factor, which is basically what a cytokine is. Notice it's a granulocyte colony, and then there's a granulocyte monocyte colony stimulating factor. Okay, so those are just first cytokines that you've seen a name for, other than that CMKL12 that we talked about earlier. Okay. All right. So extramedullary hematopoiesis. All right. So when blood is formed in the bone marrow, it is medullary hematopoiesis. Okay. So extramedullary means what? Not made in the bone marrow. It's outside. Extra means outside. Okay, exit, right? So extramedullary hematopoiesis is two places. That clock ain't right, is it? It's not 12, is it? Okay. I knew I've been talking a long time, but I didn't think I've been talking that long. So basically you have three organs that do this in our body, okay? The thymus gland, the liver, and the spleen, okay? So those are the ones that we want to talk about today, okay? Now listen, when I say thymus gland, also mean lymph nodes, okay? Because the thymus, the lymphatic system, and the thymus gland are all connected to each other. So we actually concentrate on the lymph nodes more than the thymus gland. Why? Why do we want to concentrate on lymph nodes, not the thymus gland? Yeah, but your thymus gland's on your body too.
Doesn't your thymus gland eventually stop producing that?
What does it do?
Get older?
Yeah, you're right. You're exactly right. It atrophies, right? It shrinks. So by the time you're my age and even older, you don't have a thymus gland. So can a thymus gland make you some more cells? Nope, not really. It can help a little bit because there'll still be a few cells left of that tissue, but there's not enough to talk about it. So then we have to move to the lymph nodes, okay? Because basically what happens is the thymocytes, which are basically immature lymphocytes, they're in the thymus gland while you're young, but then when you get older and it starts atrophying, they have to go somewhere else. And so they go to the what? Lymph nodes. And so then those lymph nodes become the site of extramedullary hematopoiesis. Okay? All right. But the two big ones that we want to talk about are spleen and liver. Okay? So lymphopoietic. Okay? So if hematopoietic is the formation of blood, what is lymphopoietic? Huh? Formation of what? Lymphocytes. Somebody said it over. Who said that? Atta girl, Chloe. Lymphocytes. Lymphopoietic or lymphopoiesis. Okay? Is the formation of lymphocytes. Okay? T cells and B cells. Also, we have natural killer cells that are also classified as lymphocytes. Okay? So these all take place in the thymus, okay, until the thymus disappears, and then it kind of starts taking over in the lymph nodes. Okay? Basically, you have what's called a medulla and a cortex of the thymus gland. So this is really, really important for kids. Okay? So you guys are not old enough. Some of y'all may be, but some of y'all are about my age. A couple of you in here are my age or close. All right? So when I was a kid, leukemia, if you got leukemia, you died. You know? That was just how it was. There was no treatments. Hardly anybody ever survived leukemia. Okay? So it was like a fear. You know, you get a bruise and mom and dad freaked out, you know, that you had leukemia. Okay? But there was this one kid in America that didn't have a lymphatic system, didn't have an immune system at all. And so they put this kid in a bubble, and like he lived in a bubble. And they made a movie about him, The Boy in the Bubble. If you don't believe me, look it up. Okay? Until this kid reached about 18, and then he decided he didn't want to live in a bubble no more, and he got out, and guess what? He died because he had no immune system at all. Died from an infection, just what they were trying to prevent. Okay? So the reason that Junior has a huge thymus gland when they're born is because they ultimately have no immune system. Okay? So they have to have this organ to help them to form their immune system. If they don't have it, they will never survive. Okay? Because they can't produce these lymphocytes, which is the main things. You can produce, neutrophils, and you can produce eosinophils and basophils. But guess what? They don't help you against viruses and these kind of things. They only help you if you get bacterial infections or fungal infections, okay? Or parasite infections, all right? We live in America. We don't get many parasite infections. I guess if you live in Botswana or somewhere, where you're drinking from the same hippo-infested water, you might be glad you got your EOs, right? And we're glad we have them too, but we're not as thrilled about our EOs as we are our lymphocytes. We like those guys, and we want them to be strong, okay? We can't be strong if we don't have a thymus gland to start with when we're born, okay? Because this is where they basically form. They form in the bone marrow, but then they mature in the thymus gland and the lymph nodes as we go, okay? Y'all know what a dendritic cell is? Now, okay, there's two forms of dendritic cells. Probably in 3rd and 4th grade, you learned of one type of dendritic cell when you were studying the brain and the central nervous system, okay? There's a dendritic end of a nerve cell. Y'all remember that? Axon and dendrites, okay? Y'all remember all that? Well, this is not that cell, okay? This is not a nerve cell. This is actually a specialized white cell that's called a dendritic cell. It's a narc cell, okay? It runs around telling on everybody, okay? It basically exposes like T lymphocytes and B lymphocytes to the foreign antigen, okay? Sometimes it'll even pick it up and take it to them and say, hey, get a load of this guy. And then they become immune competent to that particular antigen. Does that make sense to you? So when we talk about dendritic cells, we're not talking about your nerve cells, that part of your nerve cell. We're talking about narcs. Just always remember they're snitches, okay? They tattle. They expose things to antigens. And y'all talk about this in immunology in the summer, okay? But I can introduce you to a little bit of it now, all right? So T lymphocytes supplies immunocompetent. What does immunocompetent mean? What's immunocompetent mean? Well, what's competent mean? Means I'm ready, right? I'm competent to whoop your ass. You know, I am ready to. Don't mean I can, but I'm ready to. You know what I mean? Immunocompetent means it's ready to do what? Come on, guys, crap.
Fight off disease.
Right, it's ready to do a immune response, isn't it? Right? Immunocompetent means I'm ready for my immune system to do what it needs to do. In other words, these are cells that are ready to be exposed to a foreign antigen. Okay? Because we have immunocompetent cells, but we also have immunoincompetent cells, also known as naive cells. Okay? Naive cells, they ain't ready yet, you know? They're not ready to fight off something yet, okay? But now when they get exposed to something, then they become immunocompetent. Does that make sense? Okay? Immunocompetent cells are how your vaccines work, guys. Okay? We shot you with a dead virus when you were a baby and you didn't have no choice in the matter. and we fixed you up to produce immunocompetent cells. That way, when you're 15 and you get exposed to measles, okay, your body has a cell in there that says, I've seen that crap before. You know what I mean? Immunocompetent cells are the 40-year-old staff sergeant that's been in the military since he was 18. You know? He's seen war. He's seen peace. He knows what's going to happen. Okay? You get these new recruits that ain't never been in a battle, and they're over there shaking in their knees and in their britches because they've never been shot at, right? Those are naive cells. They're not sure. They're there, and they're willing to do the work, but they ain't real sure because the bullets hadn't started flying yet. Does that make sense? Immunocompetent cells, they've had bullets shot at them before, and they've absorbed them, and they've said, piss on it. I ain't scared. You know, I've got what it takes to fix it, you know? And so then when they get exposed to it again, they're ready to produce an antibody, which is usually the B cells that do this, okay? T cells and T4s and T8s, they're the same way. They just use a different method besides antibodies. They're cell mediated. They'll like engulf them, okay? And phagocytize them just like neutrophils and macrophages do. They just do it in a different method, okay? As we go. All right. Well developed at birth, talking about that thymus gland. Okay. Continues to increase until puberty. And then puberty changes everything for us and humans, if y'all hadn't noticed that. Thymus gland, basically the theory is that once we reach puberty, there's a little bit less growth hormone being produced. We still produce some. Because, you know, boys hit puberty about 13 and they're usually little short, squeaky kids. I just hit puberty and stayed the same height. You know, I did grow a little bit because if y'all don't see me in 7th grade, I look like a little midget. I was about this tall, probably in 7th grade. But I grew a little bit, but not much. Like my sons, though, both my sons went from this to this in like 2 years, you know. But eventually that growth hormone begins to slack back, okay? So like you don't see many 25-year-olds growing 2 inches, you know? If they do, they got a freaking problem. They're putting a needle in their butt or something, you know? Or they got a pituitary problem or a hypothalamus or something, okay? Because usually they don't grow. And so the theory is that due to a decrease in growth hormone, this stimulates the thymus gland to begin to atrophy and not be there anymore, okay? You start getting the testosterone and the estrogen and those hormones being produced, okay? So like I said, like my age, probably, I don't have probably a thymus. You probably would have a hard time finding even a little bit of thymus tissue in my body, okay? But definitely by the time you're probably 50, okay? Another thing, too, is characterized by reduced expression of the FOXN1. See, I didn't even know there was a FOXN1 when I graduated school, you know, because this is a gene. It's part of a gene. When you talk about expression of, they're talking about genes, whether they're turned on or turned off, okay? So this plays a role in producing this, okay? This is what the thymus gland kind of looks like, or like a section of the thymus gland. It's kind of showing you the medulla and the cortex and what's happening in these particular regions. So just kind of study this as you go, okay? Spleen. Any questions about the thymus? That's pretty much all you need to know as an MLT, okay? Know that the thymus gland plays a role in lymphopoiesis. It helps to form the lymphocytes, mature the lymphocytes, T cells and B cells to an extent, okay? Most of your B cells are actually in lymph nodes too. That's the reason they're actually called thymus gland or T cells, is because of they're called thymocytes for a little while when they're in the thymus. Just FYI, okay? But B cells actually form in the bone marrow, and some of them stay in the bone marrow and form plasma cells. And then plasma cells turn into antibodies, okay? But the majority of the B cells do go to your lymph nodes, and that's where they mature, okay? So the spleen, talk about the spleen. Located in the upper left quadrant, so you got to remember anatomical position. This would be my upper left quadrant, right in this area here. So this is where my spleen's at, so don't hit me in the spleen, all right? I want to be hitting the spleen, all right? It's not essential for the human life. Did you know that? I actually like to see people's blood that have had a splenectomy. And the reason is because your spleen plays a role in producing cells, but it also, remember how we talked about hematopoiesis is not just the production of cells, but it's the production, the maturation, and the destruction, okay? So when we talk about hematopoiesis with the spleen, it can actually help you produce cells, but its main job is destruction. Really, when it gets right down to it, okay? And it does it by a couple of ways. It does what's called culling. Y'all deer hunt? Nobody deer hunts? You deer hunt. So y'all cull? Do y'all cull deer every now and then? So why do you cull a deer?
Well, to keep them from taking over.
That's it, from passing their genes on, right? Because they're not good deer, right? They don't have the horns that you're looking for, okay? May not have the size you're looking for in a body, okay? So when they get a little older, or maybe just don't have the genetics, you cull them. Guess what? All your red cells aren't made the same. You know what I'm saying? Red cells get old. They can't perform, okay? I laugh at my son all the time because he's 15 and he's getting that, extra boosts of testosterone. And, of course, I'm not as much now that I've been sick because I just don't got the energy anymore. But he used to, I'd fight with him all the time. I'd wrestle around with him and everything else. And there for a little while, I could take him pretty good. It was almost like just one hand, I could push one hand and hold him back, or hold him down with one hand or whatever. But lately, I've been noticing, even before I got sick, I was having to put a little bit more effort into it than what I really wanted to. You know what I'm saying? And you know the difference? Because I'm beginning to senesce. What does that mean, Brian? Getting old. Brian, I'm getting old. I'm beginning to senesce. There's a term in science. It's called senesce. And you see, senescence is what we say. Those cells are getting old. They're getting older. Okay? I tell them all the time, I said, now listen, I don't know that I could play a whole football game. I said, but I give you hell for about one series. 4 downs, I can whoop your butt, After those 4 downs, though, I'd probably have to be on the sideline with oxygen. Please help me, God. So our cells do this, They're not as efficient the older they get. So about every 120 days, we get a turnover of cells. We usually don't lose more than about 1% at a time. But those cells have senesced. They're not as good at doing what they should do, okay? So our blood system, talking about this earlier, all right? So you're going to have to just imagine with me, because I'm not a very good drawer. I'm going to have to hire my oldest one to come and do my art for me. Okay? So, think about it like this. All right? So let's say this is your spleen. Okay? And your spleen, of course, is going to have arteries coming in and veins and, of course, everything. Actually, about, I think it's 5% of your blood volume at any given time is going through your spleen. That's the reason you can bleed to death really quick from a ruptured spleen. Okay? So you got these vessels coming in. And when they come in, they can go 2 different ways. Okay? There's a system of vessels that basically will run them through one side and then through the other. Okay? This is a quick system. Right? This is the direction that the blood prefers to go through. Okay? But what happens is there's another system where the blood comes through one of those sinuses. I don't know if that's exactly how you spell sinus, but it's close. Sinus. So what did we say about a sinus? It's an open system now, right? So instead of the blood flowing, through here in that closed system, and going out, it can go in here in this open system and kind of just get spread out. Okay? But here's the problem. Now, we would like for our blood to be able to go through this all the time. Okay? And here's why. Because in this system, it's a slow system. Okay? So if I've got a slow system, I've got white cells in there, I've got red blood cells, I've got platelets, they're all going through this little system. Okay? Now, we said that red cells, in general, they do anaerobic respiration. But white blood cells, they don't. They use aerobic respiration. So they need oxygen. Okay? So what happens in this area, in these sinuses, all the white cells that are in there, whether it's monocytes, EOs, whatever, as they're going through, they suck up all the oxygen. And then they head on out. Okay? So what it does, it creates what's called a hypoxic environment. Okay? So not only does it create a hypoxic environment, but because they are doing cellular respiration, even though even the red cells are doing anaerobic respiration, what are they producing? Remember? I told you when you do anaerobic respiration, you produce lactic acid. Well, guess what? You got all this lactic acid in here, so now we have a hypoxic plus acidic environment. So it's not a real place you want to hang out too long, right? So what it does, or what's really cool about it, is you've got these big macrophages that kind of float around in this system. And they sit there and they keep an eye out on the red cells. And if those red cells begin changing shapes or doing weird shit because they're in a hypoxic environment, it says, you're not fit to survive. And it goes in and eats them. And gets rid of them. Out. Okay? Another thing, and this is called coiling. Alright? Another thing these little cool macrophages can do, they can do this thing called pitting. Okay? So what happens is they get put in this environment, alright? And say they're in that hypoxic acidic environment, let's say their hemoglobin, which is a protein, okay? Proteins are really bad about changing shape, denaturing. You ever heard that term? They denature in acidic and hypoxic environments, okay? Because they like to be in a certain environment, and when that environment changes, they don't work right well together. So what happens inside the red cell, hemoglobin's bad about doing this. It will precipitate, and we get what's called called Heinz bodies. Heinz like the ketchup, or the 57 sauce. I like the 57 better than the ketchup, okay? But they get what's called Heinz bodies, which is basically precipitated hemoglobin. So these macrophages, they say, wait a minute, you can't do that. And so it jerks a piece of it out, and what happens, it forms what we call spirocytes. And so instead of them being that biconcave disc with the central power, they turn into globes. And they look just like a spear. And the reason it is because they're so old that their membrane that it jerked out over here can't rebuild itself. And so it basically just inflates and turns into a ball, a literal ball, instead of a biconcave disc, like what a red cell should be. Does that make sense? Okay? Another thing that it can do is sometimes our red cells have antigens. You know that, right? You know what antigens are? How many knows what an antigen is? Stick your head up and down if you know what an antigen is. So I'm saying 1 out of the whole class. So I'm going to say you don't know what antigens are. Okay? Antigens are proteins on the membrane. So you guys talk about, when you were in biology, we talked about a biphospholipid layer. Remember y'all talked about that in biology? Okay? So another thing in biology we didn't talk about is sometimes there are special proteins that are embedded along that membrane. Sometimes they stick out and they've got shapes to them, you know, all these different things. Okay? Those proteins are antigens. Okay? For like us, here's something simple. How many of you are O blood type? Should be about all of y'all, just about. Okay? How many of you are A? That ought to be the next. How many of you don't know the blood type? Holy shit! Y'all guys not like don't make blood and stuff. We need to do y'all's blood types one day, so at least you know what you got. Okay, so the majority of the people in America are O. Okay, what that means is there's 2 antigens in the ABO group, called the ABO blood group. Okay, they are actually A and B. That's the only 2 antigens that there really are. If you're O, you don't have either one of them. You don't have A or B antigen. Okay, so you don't have that protein on your cells. All right. I'm saying all this to say this, okay? Sometimes in our body, usually not our blood groups, but some other antigens, an antibody can attach to them, okay? It can attach to those antigens. And when an antibody and an antigen attach, they form what's called a complex, okay? And it weighs the cell down because usually it attaches to a bunch of complexes on that particular cell. So guess what? It's not as efficient anymore. So these macrophages have the ability to see these complexes. And what they'll do is they'll go in and they'll grab that complex and they'll jerk it off. And what happens is they form this real cool cell that looks like that. And it's called a helmet cell. It's called a helmet cell because, you know, the old football helmets, you know, like when they was real men playing football, and wore those leather, come down, all they had was the flaps. They look like that. Okay? But essentially it's because they've torn, those macrophages have torn those complexes off of the cell. And so they form what's called helmet cells. Okay? Those helmet cells and spherocytes are essentially can happen the same way. Spherocytes can do the same thing by pulling those antibodies, that antigen antibody complex off of them too. And they just can't fix the membrane. They're old. And they can't fix the membrane anymore, so they swell up like a sphere. Okay? Matter of fact, this ain't going to make a lot of sense to you guys, but it will eventually. You get in blood bank in the fall. When I was a tech, or when I was a student, I was in clinicals. We had, I was doing my clinicals at a hospital that had kids. We had babies, and had a baby that was going really, really bad, and we couldn't figure out what was going on with it. The ABO wasn't incompatible, anything like that. That was normal, the cause of these kids going bad. So I was screwing around in the morning, messing around, just playing, because I didn't have nothing else to do. And one of the DATs, which is a direct antiglobulin test, which tests to see if antibodies have attached to red cells. Okay? So I was messing around with just that. I was playing. And instead of doing it at 37 degrees, I just did it at room temperature to see what it would do. You know, because I mean, it wasn't a real patient, or was a real patient, but I wasn't reporting it. So I was just goofing off, you know, wasting time. Well, it ended up being positive, which it shouldn't have, because when they did it at 37 degrees the night before, it was negative. Okay? So essentially what it was saying it was reacting at room temperature or cold because it had come straight out of the refrigerator. And what it did, it told them that all cord bloods have what's called I think it's little I, which is a certain antigen on the cord blood red cells, okay? And mom, after we did this and figured it out, we called the pediatrician and told him what was going on. He told me, he said, well, go pull a slide on the baby. And I said, okay. So I went and got the morning CBC on the baby, and it was full of spherocytes. So that baby's spleen was working good enough to pull out those antibody antigen complexes. And so it was causing what we call spherocytosis is what it was causing. And the baby was going bad. So it was popping its cells, essentially what was happening. Those spherocytes can't last long, and so then they'll hemolyze or lyse and release the hemoglobin, and that was what would happen. The baby's bilirubin was going up because of the breakdown of all the hemoglobin. And so we found it and figured out that mom had little I, and she was destroying the baby cells, her antibodies during the birth, because it was sensing it as foreign. Okay? All right. So that's what the spleen does. So basically, we talked about all this. You can live without a spleen, but it's not good. It drops your immune system. People who have a splenectomy, you look at their blood smears. We hadn't talked about morphology yet, but I did. How many of you looked at the PowerPoint on RBCs yesterday? Absolutely none of you. Damn, you did? So you saw all those shapes, you know, like acanthocytes and like helmet cells and spherocytes. Okay, you didn't look at that one then. There's another PowerPoint that's the WBC, but then there's one that's an RBC one, okay? You need to look at that soon, okay? We're going to talk about that stuff next week when we start talking about the erythrocyte in general, but it doesn't hurt you to go ahead and look at it. But what I'm getting at is your spleen's pretty efficient in pulling those old raggedy cells out that don't function like they should, okay? So if you take a person who doesn't have a spleen, they don't get pulled out. The liver can do it, but it doesn't do as good of a job as the spleen, okay? And so one of the artifacts, what we call an artifact because it's not a real symptom from a splenectomy, is that you see all this trash on their slides. You'll see red cells that are all shapes and sizes and different things because they didn't get pulled out by the spleen. Does that make sense? Okay? So if you ever see that as a tech, when you get older and get into the lab, that ought to be your first thought, is this person doesn't have a spleen, you know? And it's a good thing to note because you can pick up the phone and call the doctor and say, hey, they had a splenectomy. If the doctor says no, then you tell him, say, look, I've got, you know, we would use science terms. We would say we have, 4+ anisiocytosis and poikilocytosis on this slide. And what that lets them know is that their spleen's not working like it should. And they can actually do what's called an autosplenectomy. It means the spleen's still in their body, but it ain't working. It's died. It's not doing what it needs to do. Does that make sense? And I'll talk about that in just a second. Okay? Colon pitting. Talked about that. That's that slow transit pathway. That's where what we were talking about with the open system, where the sinuses, where the blood goes in. Know that the spleen has red pulp and white pulp. Okay, those are important things. That's what there's cords that go in, and these cords are kind of where those things, those macrophages hang out. Another thing you need to know about spleen is it actually stores platelets. So like when the platelets break off the bone marrow from the megakaryocyte, they go into the blood system. You don't need them all in there at one time. So they go to the spleen as a storage area, okay? So what will happen a lot of times is that their spleen may go bad, okay? Like we're talking about the autosplenectomy, and then you'll see this huge increase in their platelet count all of a sudden. And what it is, their body kicked all those platelets out of the spleen before it died. Does that make sense? And so you'll see that a lot of times as you go. All right? Yes, ma'am.
So why when you have mono, they tell you no physical activity because of your spleen?
Okay, so one of the things that happens because your spleen's job is to filter, and we've got all these macrophages and stuff in here,
And like, we're doing one, like but there's like thousands of these little things going on in their body. So what happens is you overstimulate your spleen. When you get sick with mono or anything, there's actually lots of things that can cause splenomegaly, is what the term is. And you get an enlarged spleen. But mono is one of the big ones that does this. And so what happens is essentially blood infiltrates more, you get more blood flow because you're sick, okay? And so when it gets in there, the blood it begins to swell. And not only that, those macrophages are overacting, they're overreactive, and so that causes inflammation to occur. So you get fluid other than just your blood, like lymphatic fluid and serous fluid, and those kind of, not serous, but it is technically serous, visceral fluid, okay? It all goes into your spleen, and it causes it to swell. And so normally, you have protection, okay? But like if you go out and play football, which is what I did, like a moron, because I didn't know. Nobody ever told me. I got mono when I was in the 9th grade. I was sick for a week. And, I got sick on like Wednesday, Tuesday night. I played football. And I was a running back. I was a fullback. So I blocked and got tackled. And I was recovering from mono. And, nobody ever said, hey, you don't need to play football because your spleen's large. You can bleed to death and die. And my parents wasn't in medical at that time, so we didn't know. Just didn't know. But yeah, does stupid things. But yeah, that's why. It's because it just gets so overactive that it basically begins to enlarge. But splenomegaly is a big thing. We'll talk about it in leukemias. That's one of the signs of leukemias also. You start seeing all these splenomegaly. Hepatomegaly, which is your liver, gets enlarged also. And one of the reasons they do that too is because a lot of times people who get leukemia, their bone marrow begins to become hypoplasic, and so it doesn't make those cells. So they kick in and start trying to do extramedullary hematopoiesis, like what we're talking about. And plus, there's such an increase in number of cells, they're also destroying cells, not to mention trying to produce cells. And so it basically just, it's like, if you work out, what do? You get big, right? And that's what happens with your spleen and your liver. And they just get larger and larger until eventually they blow if you're not careful or quit working because they're overreacting. Lymph nodes, I'm just going to let you guys read this. Yes, ma'am. Huh? No, go ahead. I got all day. I don't care. You're ready to go. That's what you was going to say. Chloe, I can't believe that. This is college, baby. Get up and go. You don't have to ask me to go. Just go if you want to go. Read about the lymph nodes. Very important. This is where the B cells basically mature, the germinal centers and all that. And like I said, we'll go into more detail about T cells and B cells later on. This is just intro, kind of giving you the tissue and that kind of stuff. So don't think when you leave today, you're going to know everything there is to know about B cells and T cells, because you're not. We're going to individually talk about those a little further in, okay? And I think that's it, really. Do mention MALT. MALT is lymphoid tissue that's not associated with like the thymus and the lymph nodes themselves. So like on your intestines, those kind of places. Peyer's patches in the intestine. I don't know if you've ever heard of that or not. Tonsils in the appendix are also those. Enlarged lymph nodes is known as lymphadenopathy, is what that term is. Okay? All right. We'll stop right there. That's chapter 4. All right. See you guys tomorrow. Tomorrow, right? We'll do it. Yeah. Everybody look at those cells. Don't forget your atlas. Make sure you bring your